Welcome back, all. This is the Straight from a Scientist podcast. We are back here in 2022. Took 2021 off in the back half of 2020 because of coronavirus and some other things. Most notably, I graduated, so I'm actually now Dr. Connor Wander. Got my PhD from UNC Chapel Hill, and I have way more updates to share with you. But first, let's get into the episode. This is an episode with Dr. Esteban Oryarzabal, also newly minted. He is now at Charles River Labs, but this is a study done at UNC Chapel Hill. It's a great neuroimaging study all about the locus coeruleus and the default mode network and what that could mean for attention and our understanding of just the basic functions of the brain when you're task switching or just moving around from trains of thought day to day. So we had a really great, it gets technical, but it's fascinating, and we go surface level all the way down deep into the weeds. So stick with it, and there's some great visuals for those of you listening purely on Spotify or iTunes. Do check out the YouTube episode, as I think it helps a lot to have those visuals there and just understand what these different networks are doing when they're all tied together. So some housekeeping stuff, I guess I should say, a few updates, and I will do some more updates, just kind of what's been going on in the past year and a half. Julian is now in graduate school, and throughout the duration of the back half of 2020 and 2021, I had actually switched from podcasts and was working on these web science games, which are still under development, and some great help from some student interns at Apex High School in North Carolina. So still very local there on the East Coast, and we're working with them. These web science games are perfect for students, perfect for anyone who doesn't really fully understand virology, coronavirus. Shout out to Calvin Yeager for helping me with the coronavirus games, which are extensive but are a lot of fun, and see if you can get to the really interactive levels on there. We also have 2 other neuroscience games, still all in development. Definitely best to work on your desktop, but try them out. Let us know what you think, and definitely share them with others. So with that, I will get into the episode. Lots more coming from us, restarting Straight from a Scientist as I've gotten settled here in California, and just kind of lots more updates to come. Enjoy your year, stay safe, and let's get to it. Welcome, everyone. Connor Wander here with Dr. Esteban Oryarzabal, I should say. Congrats on the recent graduation. We're going to talk about Esteban's recent publication in bioarchives called Chemogenetic Stimulation of Tonic Locus Coeruleus Activity Strengthens the Default Mode Network. Now, I know that's a huge word string. We're going to break that down, and we're going to talk about what Esteban's been doing post-PhD, as well as maybe a little bit about what I've been up to, because I know you all deserve an update. So thank you, Esteban, so much for joining in. I know you've been busy.
Yeah. No, not a problem, Connor. Thanks for inviting me for this. Yeah, it's been great.
Yeah, I'm really looking forward to this, because the locus coeruleus to me is this kind of black box of the brainstem. So it's this deep part of the brain, and it's very hard to study. First off, we know it's important, but we don't exactly know why. And I think your paper does a really good job at like starting to dissect like what exactly is going on in the locus coeruleus. So before we get into too much about the locus coeruleus, I want to talk about this default mode network right at the end. A lot of people refer to the default mode network, or the DMN, a lot in literature, but it's kind of nebulous. Like, what does it actually do? What is the purpose of it?
Sure. Absolutely. So I guess one good way to really start about understanding the default mode network is sort of how it was found. So when fMRI was really in its infancy. So one of the cool things about functional magnetic resonance imaging is that it first really started in humans. And so one of the first things that we utilized fMRI for was we would put a human subject in the scanner and have them perform a certain task. And so that kind of gave us an idea of what different brain, where exactly behaviors are involved in certain brain regions. So we could actually start localizing that.
So that before we go too deep into the weeds, fMRI, functional magnetic resonance imaging. So this is a way to scan the brain and look at what parts are active without like putting an electrode into the brain or hurting anyone in any way, right?
Exactly. Yeah. So it uses what we're actually scanning. So the bulk of human scans is done using a modality called BOLD, which is blood oxygen level dependent. And so what that means is when your hemoglobin actually releases its oxygen, those iron particles on the heme group now kind of change the magnetic field. And so that's what we're actually imaging, is when that action of a oxygenated blood becomes deoxygenated. And so that kind of one of the beautiful things is there's this relationship between the vasculature and neuronal activity. And so it's a dogma that it's called neurovascular coupling. And so the dogma is that when you have a lot of activity in a brain region, more of the end blood is going to be deoxygenated. And so you're going to have this change in particular. And so that's kind of the kind of the measure that we have with fMRI. In our paper, we actually used a different modality, which is called CVD weighted. And so what we're doing is we're actually putting iron oxide nanoparticles in the blood. And so that actually gives us higher resolutions. And so we're actually measuring the total cerebral blood volume. And so instead of just having only the deoxygenated signal, we have like the whole blood volume signal. And that's really important when you're working with really small animals, because you need to have as large of a signal and signal change as you could possibly detect. But so let's go back to the default mode in general. So, they were out there and they were having people like do like a finger tap or something like that. And one of the things that they were finding was that there was a brain region that was always active in the passive control condition. So the times in which people weren't doing anything. And they kept popping up these networks in particular. And so that's really how they found this particular network, the default mode network. And one of the other things that you should note is that historically, we always thought that the brain, everyone talks about this 20% of our glucose is consumed by the brain, yet it only makes up like 2% of the body weight. So we always thought that 20% was going to when we were actually engaged in like an executive function or like sensory type of relays or things like that. But what they actually found out was that most of the glycolysis in the brain occurs in this resting period when we're not really doing anything. And so it's actually feeding primarily into this default mode network. And so what we know about it is that it's active when our brains are not really engaged in sensory processes of salient stimuli or cognitive tasks. And so we kind of think that it's kind of involved in some central functional organization of the brain. And as you can imagine, it could also become uniquely vulnerable to dysfunction, and that could actually cripple many systems of the brain in general. And so, so one of the functions is this idea of self-referential kind of thinking, or kind of mentally processing and imagining past events or upcoming events or future events. And so, for instance,
so daydreaming, one of the,
right? Yeah, exactly. Daydreaming, all that stuff. So one of the coolest studies that was done was actually with Buddhist monks. And so people who meditate a lot and have that internal kind of reflection tremendously. And when you actually put a Buddhist monk in there, they have the most robust default mode networks. So how you could view it is this way. When your default mode network is activated, you're actively suppressing all sensory stimuli. So someone that could dissociate and like get into, almost like a matrix's zone that it's just like, a white box, sterile box or something like that. If you could mentally achieve that, that means you have this tremendous control from the default mode network to your sensory stimuli. Yeah. And so that's one other thing that we do know that is actually really involved. There's two other networks that you may have heard of. One that is called the Dorsal attention network. And so that one consists of regions that really are involved and engaged when we have external oriented attention towards like salient kind of activity in general.
So we're busy doing something and like very focused on that task,
what lets us like focus in, like it's what lets us reorient our mind to outer kind of stimuli in general. So there's two. There's that one and the salience network. So both of those networks really are kind of consistent in a lot of ways. But one of the cool things about it is when those are activated, the default mode network is suppressed. And so what we don't really know is actually the temporal order of how that switching happens from one area to another. And that's actually kind of one of the reasons we entered this project with the locus coeruleus, is because we're really trying to find out what are kind of the neuromolecule signals that could be involved in these type of gating between networks in general.
And I think this whole network switching is, I would say, impaired or at least implicated in a variety of diseases, right? So
yeah, tremendous. So imagine someone with attention deficit disorder.
Right.
So part of the reason, so we talked about the Buddhist monks and being able to zone into like a blank room almost with no outer stimuli whatsoever. Someone with attention deficit disorder is the opposite. So much of that outer signal is coming in, or people with autism, like they have a tremendous amount of sensory stimulation coming in. And so one of the beautiful things in particular is, it's kind of, the idea is if you can find a way to, so because we don't really know the mechanisms that govern how to keep those distinct, if you could find a way to drug something like that and actually kind of allow those networks to actually be more anti-correlated to one another, you could start focusing people's attention. You could start, getting them into a way that their processes, or their brain functions and processes, are more distributed in the way they're intended to be distributed, I should say. Yeah.
Or more controllable,
or more
by
controllable.
the person themselves.
Exactly.
Yeah,
instead
absolutely.
of a pathological condition. Yep.
Yeah, and I think if we think of a lot of the medications for attention deficit disorder, for example, it's if anyone has ever used them or talked to someone who uses them, it's almost like they're forced into one of these attentive states by these drugs, and they can't really relax. I think that's one of the side effects, is like you're really tense and hyper focused, and you can't just like kick back if you've taken the medication.
Yeah, and one of those things, one of the key things in particular, so obviously there's the noradrenergic version of it, but there's also the side that hits dopamine and serotonin. So there's different strategies at targeting attention. The dilemma is when you use medications like that, they're hitting typically the receptors, which are on the postsynaptic side, but we're not just selectively saying, oh, we want to target, the anterior cingulate. We're saying we're just dumping it everywhere that has a receptor. And so the problem is you start having side effects as well with all these things. And that's kind of like, that's we'll get into it a little later, but that's why models like DREAD in particular are really cool, because they are kind of looking at it in the future of how can we selectively just target one region. And we could talk about that more later on.
For sure. Yeah, I think so. Let's get into the chemistry part of it, right? Because you mentioned, we've mentioned noradrenaline, we mentioned serotonin and dopamine. So these are monoamines. And then noradrenaline, a lot of people will think, oh, adrenaline, I know that. That's like when I get scared, I'm on a roller coaster, or like for me, if I'm playing paintball or something fun. But adrenaline and noradrenaline are a little bit different. They're chemically a little bit similar, but I think there's a big distinction there, and we need to clear that up first.
Yeah. So those are those particular, the monoamines are what we call the catecholamines. So they all come out of an amino acid, tyrosine. And so as we convert that, the first step it goes to is L-DOPA. Then it could be converted to dopamine, which all of us really know. It's involved in Parkinson's disease. And then the next step in this synthesis process is norepinephrine. And then the next step is epinephrine, or noradrenaline and adrenaline.
Depending on
lately,
which
we've actually, the field has really changed the name. But those names are ones that are part of the reason we have the understanding of neurochemistry and neuroscience, because those are some of the first neurochemicals ever discovered, actually. And so, of course, adrenaline, you think about it, you're thinking about fight or flight type of responses, these like panic responses. And yes, and one of the beauty is a lot of people, they're technically called neuromodulators. So one of the things you could do is you could kind of divide the brain or neurons into like three classes. Your excitatory neurons, your inhibitory neurons, which are kind of self-evident, and your neuromodulatory neurons. And your neuromodulatory neurons really shape the response of excitatory and inhibitory neurons. So one of the ways I like to describe it is kind of like tuning a radio dial.
Perfect.
And once you get it, you look at it in a way that's like, neuromodulators really help you get it. So it's like the fidelity of the signal is like perfect. But imagine if you have a dysfunction in those systems, then you're getting noise. You're getting all this noise, and you're not really hearing the signal that you're trying to like perceive. And so that's one way I like to describe the neuromodulatory system. And it's really different in a lot of ways, because I think a lot of people, when they think of neurons, they always think of like this monosynaptic kind of model in which it's like one neuron sends a synapse and like dumps a neurotransmitter.
And like one linear chain. Yeah.
Exactly, or one linear chain. Neuromodulators are like a massive cell body, and they send these projections that go throughout like the entire brain. And they have what we call them, we call them end-pass kind of synapses. They're like these little balls that are across this projection, and each one releases neurotransmitter, or in this case, neuromodulator, norepinephrine. And so one neuron could have like 10,000 to 100,000 synapses. And so early on, when we really started thinking about norepinephrine, a lot of people just kind of put it off, because they look at it as a system that just like innervates everything equally. And they're like, well, that's just background, which in a sense it is, because it controls, when we're talking about signal fidelity, it's controlling the static, whether you have too much static, like spontaneous firing, or not. So I think that's a good way to really understand that particular system. And another thing that's really important, like you were saying with adrenaline in particular, is that it controls a lot of your physiological responses. Norepinephrine is the same way. So norepinephrine itself could like change your heart rate, blood pressure, body temperature, how you break down fat, and like kind of the amount of blood sugar that you have in your body. So it has a tremendous amount. Oh, and then of course, most importantly, it's also one of the most potent endogenous vasoconstrictors, which is something we'll talk about a little later about this paper. And then when you look at it, so that's in the peripheral system, but in the brain, it's involved in like wakefulness. So right when we wake up, that's like really the key signal that really starts getting us out of bed: arousal, alertness, kind of vigilance, these ideas of memory formations and focused attention. But they're all more like abstract type of behaviors, because part of the reason is like they're involved in the actual processing of those they're kind of that background idea that I was talking about reducing noise. That's part of what they're doing. They're really improving signal fidelity along multiple brain regions, and really saying, this area is important, this area is maybe not as important right now to be focusing on. So it's known in this idea to be an overarching system that could maybe be helping transition between different brain networks. So that's one of the coolest things about these neuromodulatory systems.
Nice. So let me know if I'm thinking about this wrong, because
Sure.
I want to compare these to like real life events, right? So let's say I'm mountain biking or whatever, right? Sure. And like I'm zoning out for a bit. I'm like on a flat piece of ground, probably switching to default mode network a little bit. Yeah. Then I start going back down a very steep hill, and there's a bunch of rocks and stuff like that. When those start coming in, my noradrenaline levels start coming up. My adrenaline levels start coming up if I feel like I'm about to fall or something like that. Yeah. And the brain is really just keyed in on, okay, what's the next object I'm about to smack into potentially, right?
Absolutely. Okay. And that is the system. So the locus coeruleus in general. So the noradrenergic system, we have kind of these clusters of norepinephrine producing neurons all over our pons and medulla of our brainstem. So there's lower like 7 or 8 of them, depending on species. But so the locus coeruleus is our primary one that we think about because it's the largest of all of them, and it innervates most of like the frontal and cortical areas, hippocampus. There's also some subcortical regions as well.
Let's start screen sharing here.
Yeah, absolutely. So there's a good number one, really kind of indicates that. So it's really most of your brain. There's some areas in subcortical space that really aren't covered so much. And another area that has really little innervations is actually the striatum. So striatum tends to be a really good control region to actually pick for when you're doing activation like this. So, but I guess one of the cool things in particular is that when you activate that, so we have what's called a reverse U function. So one thing you need to know about these systems is most of these neuromodulatory systems are tonic firing. So that just means that they are, when we are awake, they are just kind of like a metronome, boom, fire, boom, fire, boom, fire. And so they're kind of bathing our brains in norepinephrine, okay? And what happens is as we start increasing the firing rate, then that's where we could actually start getting more into a focus state. So kind of the third image right there is kind of gives you an idea of what that means. So it's called a reverse U function. And so once you get to this optimal level of enhanced firing rates, what happens is we start getting into these bursts of phasic firing. So we get these like really fast firing that happens in that kind of zone. And this was all work done by the Aston Jones group, which is a really, really famous group in norepinephrine studies. And so, but if we go beyond that, so that area, that focus is like, let's say, I remember once I was in a car accident, and I remember the second it really happened, it was like everything went into like slow motion. And you remember like every little aspect, and you're like, how does your brain get into such a like focused state? That's exactly how it is. And so when you go into the other aspect, if you go too high of firing, so people that have anxiety tend to actually fire way beyond that focus state. And so then your mind actually goes into more of a like non-attentive kind of dysfunctional state. So so it goes, that's why it's called a reverse U function, because it's like optimal and then the two corners are lower, and so they're not optimal in particular.
Okay,
so I think that's a good way to really view it, and that's actually a good way to really describe why that switch could happen, where you could go into this like unfocused state and then jump into this extremely focused state. And so like you were mentioning, the unfocused state, your default mode network may be more active, and when you're in the other one, you have that very high separation between the two networks.
So it's not necessarily about a Goldilocks zone so much as we want to be flexible. We want to be able
Exactly.
to adapt to whatever the situation demands, right?
Exactly. Exactly.
So it is really all about networks in that sense. But I think there's probably some pharmacological tools, so some either drug tools or research tools that we can use to modulate this, right? And this is exactly what the paper got down to. So
Absolutely.
you were basically activating the locus ceruleus and then seeing which networks were changed. So just to recap for the audience, because we do have great visuals here. Thank you, Esteban, for this.
You're welcome.
So here's the innervation that you were talking about of the locus ceruleus and the norepinephrine that gets jumped. You can really see this is all throughout the brain, right?
Yeah.
We also know that these chemicals have receptors, which are basically these a lot of people say like locks or keys, which is an oversimplification.
Sure.
But if norepinephrine were the key and then basically the receptor were the lock, and I often say like, don't think of it as a door, which is just binary, but think of it as like a car where if you turn the key, a lot of different things can happen. Like the lights can come on, or, you know, maybe in some neurons, the lights come on, but then the sunroof opens too, or something like that. Right.
Of course.
And then,
but also you should note that in this scenario, if you are, let's say, drugging it with something that is more of a, what we call them promiscuous ligands, in which it means that basically, so this is like these are the adrenergic receptors that respond to it. But if you were to look at those other catecholamines that we talked about earlier, dopamine and then adrenaline, obviously because the structures of these compounds are so similar from one another, the receptors can actually bind to each of these things. And so that's where it gets like kind of tricky when you're actually kind of trying to target some of these receptors, because it's like you're sending a universal key that's like hitting all of these receptors in your analogy right there.
And the trunk opens when you're on the highway.
Yeah, exactly. Absolutely.
Yeah, that would be an issue, right?
Yeah.
Okay, I love it. Yeah, and overall, I think the goal is to find out how to promote not only the proper brain states, but proper brain states for the fitting time, right? So that's a big ask, definitely. But let's get into how you actually did this. So we know the locus ceruleus is key to this. How do we turn on a brain at will in rodents? How does that work?
Okay, well, so I guess one of the things that we really have to talk about is kind of the advances that have been happening in neuroscience right now. So one of the beautiful things with the Brain Initiative is it allowed neuroscientists to go back. So in the past, NIH really funded only models with diseases. And so we no longer were funding what is called basic science. It's really the understanding of how neurons really work. And the Brain Initiative allowed us to go back and rethink and redesign the whole aspect of how we study the brain. And from that came some certain tools. So one is optogenetics. So that is putting in, basically these are ion channels that can get activated with light. And so they open up and could fire a certain population of neurons. And so with the advances of making kind of genetic modifications, you could actually start putting these receptors on very selective neuronal populations. And then the other one actually was developed at UNC Chapel Hill through Brian Roth's group, which is DREAD. So these are designer receptors that are exclusively activated by designer drugs. Is what, so it's like
it's anyone looking this up. It's like, 2Ds at the end. Yeah, anyone looking this up is 2Ds at the end. Just make sure you do that in Google.
And so what's cool about this? So Brian Roth comes from a lineage where he actually just studied what are called G-coupled protein receptors, which, by the way, this alpha adrenergic and the beta adrenergic are maybe the most well-studied GPCRs in the world. They've just, their study alone has, I think, issued 3 different Nobel Prizes in our understanding of these G-coupled protein receptors. But so a lot of our neurotransmitters work through that system. And so what he did was he was looking for kind of some mutations that he could make, and he found it on an acetylcholine receptor that he can actually put in his own selective ligand to activate it that had no other GPCRs that would get activated by this. So, you know how we were talking about all the dirty drugs that we typically do are hitting multiple receptors. This is like having a pristine key. Just turn this one particular lock and it doesn't open any of the other ones. And so that's kind of a good way to look at it. And so as you could imagine, when he was really coming up with these ideas, he's thinking, how could we implement this in medicine? Could we actually go and insert these particular GPCRs in humans and start altering how we target with medication? How could we make it more selective to the specific system that's broken instead of like, you know, something beyond? So that's where chemogenetics came out of. And one of the beautiful things about chemogenetics that's a little bit different than optogenetics are driving it by light. And so that system itself really isn't one that is physiologically relevant. I shouldn't say relevant, but
Not
physiologically observed in a human brain. Whereas the GPCR type signaling is exactly what we do in our brains. And so what that means is when the ligand hits, it actually alters that activation for a pretty long period of time. So it will activate those neurons for like a very, very long period of time or suppress. There's also inhibitory versions of this receptor as well. And so that's really cool because it actually means that we could implement this type of technology in a human, and then you may be taking like 2 pills a day and it would be enough to cover this.
Yeah, I mean, I think it has such broad implications in medicine. It's already revolutionized neuroscience in terms of preclinical models. I'm really excited about the potential to use some of these systems and basically use some of the things that you've learned and you're teaching the world on humans. I mean, let's not get too ahead of ourselves, but I think the implications are really strong. So I do want to say something about optogenetics.
Sure.
The photoreceptors in your eyes actually use very similar ion channels, right?
Yeah, absolutely.
So if you can imagine optogenetics being turned on by light, that's really what like your eyes are doing if they're functioning properly. Chemogenetics, on the other hand, because they are just kind of these GPCRs and they're activated by a molecule rather than light, it's a little bit more practical, right? We don't necessarily want to be shining lights into our brain if we're adding these photoreceptors there, right? I'm happy just the only light I want to see on any neurons is on my eyes, right? I don't necessarily want an implant. However, one thing optogenetics does let us do is modulate a frequency. So you can send a pulse of light at a certain time and the neuron would respond. So I think that's really the big advantage from optogenetics to chemogenetics. Chemogenetics are just so much more simple to administer. And I would guess maybe that's why you chose chemo for this experiment.
Yeah, well, so part of the reason is actually because of our collaborators. So Patricia Jensen, so she is a collaborator from NIHS, so National Institute of Environmental Health Sciences, which is in Research Triangle Park in North Carolina. So her group actually was developing a transgenic line. And so the reason we chose in particular is because these lines are, so you don't even have to do a surgery. You don't have to go into the brain, put a vector in there to transfect something. So these animals are surgically naive. So they have no damage whatsoever in their brain. And they are selectively expressing these dread receptors in neuronal populations that, so part of it is this model hits, it's a triple transgenic model. So any cell that expresses engrailed 1 during neurodevelopment. So this is during, the animal is in utero, it's expressing this. And then after it's born, it no longer is expressing this. So this is a transcription factor that only happens in this short window during neurodevelopment. And then it also needs to express DBH, which is the protein or the enzyme that converts dopamine into norepinephrine. So it's selective for noradrenergic and also adrenergic neurons, I would say. And so what's cool is that population that express both of those is very exclusively. So if you look at this image, the pink cells. So it's almost primarily in the locus coeruleus with a few cells in the subcoeruleus. And here you could actually see this. So this is what we were talking earlier about the different adrenergic sites. And this is in mouse in particular. So we have A1, A2, A5, our subcoeruleus here, and then the locus coeruleus. And so as you could see, these cells are almost exclusively in the locus coeruleus. And what's cool is that unlike when you're doing surgery, and you could be varying just a little bit, because the reality is, every mouse is uniquely a little bit different. And so you may actually not get a consistent population. What's cool about doing it in a transgenic model like this is that it's very consistent. So you're constantly getting these particular things. It's a bit difficult because you have to actually breed a ton of animals, and only the ones that are het for like both of these particular things will really express dread. So yeah, there's just a certain population out of everything that you breed that actually will be able to be used in a study like this.
But what it gives you is a very, very clean targeting, right?
Oh, absolutely. And so that's part of the reason that, well, first, so their group not only looks at locus coeruleus, lately they've been doing some other studies using a line called HawkSP that hits some of those other sites like A5, A1, A2. And it's cool because a lot of those, locus coeruleus is pretty well studied in the realm of norepinephrine, but some of these other systems only right now are emerging, and we're really understanding their roles. So it's really cool because these models really allow very clean ways to kind of parcelate these populations. So I think that's where something like fMRI has also been a huge advantage, has been that historically we've always kind of parcelated the brain based off of certain kind of boundaries and things like that. They're actually parcelating based off of genetic, so where it comes from, where these cells stem from, which is really interesting because there seems to also be a very specific relationship with behavior on those, ontogenic, like the lineage of these particular cells, which is really cool. And it completely views neuroscience also a little bit differently, which I think is kind of shifting right now, specifically with things like fMRI, that we're now functionally parcelating brain regions that we never even thought were brain regions because they don't really fit the mold of how we parcelated things in the past.
Yeah, and it's important to note for the non neuro expertise people out there that just because a brain region is associated with a certain type of cell does not mean that's the only type of cell there, right? And so that's why these genetic tools are so important, because if you go in and you say, I want to hit this spot, well, that spot probably has half a dozen or more subtypes of neurons in it at a minimum, right? So that's like really I think the advantage of this technique is we can say definitively, okay, it's actually the noradrenergic neurons in this region, not just the locus coeruleus as a whole.
One note I should make, and this is part of the reason why in the paper we don't really say norepinephrine that much, is because a lot of these cells tend to co-release some neuropeptides with them.
Right.
And so to say that it's exclusively an effect of norepinephrine, we try to like back away, but these are norepinephrine producing neurons, but some of them will sometimes release some neuropeptides as well, which that in itself is a little bit challenging to kind of like parcelate out, because that is just physiologically how these cells package their vesicles and release.
And most neurons will co-release a lot of neuromodulators at once. So I think the devil's in the details. Finding like
GABA neurons that have excitatory. It's gotten weird now. It's like the more we study it, the more confusing it gets.
I mean, if we want to take the key analogy farther, imagine you get this envelope of keys every time someone hands you a key, and you gotta, or like one of those rings, like security guard rings in the movies, right? So I mean, that's kind of what we're dealing with here, and you got to start somewhere. So I love you mentioning those caveats, because they're important for people who are
Yeah,
trying to solve future problems.
absolutely.
Speaking of future problems, I mean, the network analysis, we got to talk about that, because
Sure,
I'm going to jump to this next slide here.
sure.
This seems like trying to find a needle in a stack of needles if you're looking for something particular, right? So how does this network analysis
Sure.
work
Sure,
in
sure,
a broad scale?
absolutely.
Yeah.
So one of the things that we know, so we're fortunate that the default mode network is not only present in humans. It's pretty much most of the mammalians, no, all of the mammalian species we've tested so far using some form of neuroimaging have a default mode network. And so this is really important. That was one of the first steps we really needed to verify. When we first started this project, there was very few papers really done with understanding how the default mode network was in rodents, let alone in mice. Primarily when I first started in the lab, we were almost exclusive doing rat, because rat is a much larger species. And so as you start imaging things that are very small and have smaller brains, you actually start having to use much larger magnets. Our magnetic field is a 9.4 Tesla. So just to give you an idea, most human scanners are 1.5 Tesla to 3 Tesla. And so we are pushing the magnetic field tremendously at that point. And then we use these little head coils that let us selectively be imaging only the signal coming out of the brain. And so we are then able to start looking at patterns. So one thing everyone really should understand is that this idea of our intrinsic functional connectivity networks. And so when we're just at any given time, if you're looking at brain activity, it doesn't matter if you're alert, drowsy, asleep, sedated, or even if you're in a non-vegetative coma, your brain is active. And what's cool is that if you look at just the activity in MRI, you start seeing kind of patterns happening. And this is typically in MRI, it's due to perfusion. The perfusion is going in certain directions, and that should be according to our neurovascular coupling dogma associated with neuronal activity. But what's cool is if you start looking at those patterns, you could actually ask, are these patterns associated with brain regions or brain networks? And yes, they are. And so these concurrently active areas are not only functionally connected, but then you could actually look at their anatomical connections and start seeing that those are indeed anatomically connected. And so we focused in on the default mode. So most people typically, when they study the default mode, will just use kind of a node extraction method where they'll put just a cursor in the retrosplenial cortex or in the cingulate cortex, and they will pull something that looks a lot like this. And so what we did was we kind of already had established what the brain regions are. So we pulled those, we manually selected those using the Allen Brain Atlas. And then, and sorry, I should mention, we did a thing called independent component analysis. So independent component analysis really allows you to find patterns. And so what we were looking for is we parcellated the brain into 100 patterns. And those patterns kind of, you could say, are functionally unique regions. And so this is a technique that's being done currently in human neuroscience. And what I was telling you before, that we're now finding regions that are different than what the anatomical areas kind of, we took that approach. We took an approach of like, what does the function of this brain tell us? Where are these particular regions that are not necessarily, associated with these rigid lines made by atlases? And so that's exactly what we did here. And one of the cool things is we did a modularity analysis, and we were able to actually parcellate the default mode into these 3 regions. So one area is the frontal region. There you can see it in yellow. So here we have anterior cingulate 2. We have these lateral orbital regions and these prelimbic and areas in general. And then we have the pink one, which is the RSC hippocampus, so the retrosplenial cortex. This has an area of the, where you're selecting right there was the posterior cingulate. And then you have these temporal areas closer to the back in the hippocampus. And then the association, which is in blue. So these are these parietal cortices. And we also have these, the auditory network. So the auditory is actually, it's interesting because in rodent, that area is activated. In human, it's more visual than it is auditory. And so there's slight differences between human and rodents in general. And obviously, one key aspect of the human is the anterior areas. That's one of the most one of the more modern evolutionary areas. And so that area in humans is extremely robust. Whereas if you look there, you see the olfactory bulbs. So in humans, our olfactory systems are like nothing. And that in a rodent makes up a huge portion of the signals, because obviously to them, it's extremely important to have a strong olfactory system. So so there's obviously anatomical differences between species as well.
So I love the distinction between functional regions versus anatomical regions, if we will, right? So for anyone who would go online and go to the Allen Brain Atlas, which I highly recommend, it's really useful for determining, okay, let's say I'm interested in this brain region. What are the types of things that are expressed there? What are the markers I can use? How do I distinguish it from the rest of the brain regions? What's special about the neurons in this population? But what you're saying is you're using these fMRI techniques. So you're basically looking at blood oxygen levels. And that's, again, a proxy for the kind of activity that's there. So if neurons are more active, they're consuming more oxygen, and that's a proxy for their activity. It's not direct, but I mean, it's the closest we can certainly get using this technology.
Absolutely, and I think, in our case, it's actually CBV weighted, but I know up here it says bold template and stuff. But yeah, we can do bold on mouse, but this is a way that we could actually increase the amplitude of changes is by just looking at whole cerebral blood volume. And that's actually what we did in this particular study. But no, it's dead on exactly what you're saying.
And then what you're saying is also that you're kind of blurring the lines between what we thought we knew about brain regions and what we know now. So did you have to draw your own? I see a lot of acronyms here that I'm not necessarily familiar with. Just we don't have to go through them.
No. So it's like no. So these are based off of. So like I said before, we didn't really draw it our own. We let the data decide, and so we did basically a blinded. When we do independent component analysis, what we were looking for are regions in those independent component analysis that fall within known regions in anatomical space that are associated with the default mode network. And so some of the reasons why you get visual and this other thing, retrosplenia, let's say in that particular scenario, is because it fell between two known. So that particular node of functionally connected cells is actually a cluster that falls in between. And so that's kind of how you get those particular type of scenarios. And one thing I think a lot of the listeners should actually know is that it's not uncommon in some brain networks that you may have a certain hemisphere have a stronger amount of signal than another one. That's actually fairly, exactly. That's fairly common. And so I think by using, let's say, just an anatomical mask, you could actually do a disservice to yourself. So one aspect of thinking about it would be, in our case, when we do functional connectivity, if you start introducing segments of the brain that actually are not in synchrony, you actually start diluting your signal. And so using the functional signal is actually a really good way, because you actually are trying to parselate only the portion of the brain that has that. So obviously in something like a human, it may actually be even more important to use, because we have much larger brains. And so that means that the voxel, which is our minimum imaging unit, can actually So let's say you have a brain region, you may actually have like 1,000 voxels in that particular brain region. But if you compare it to the rodent, you may have 3. And so it's really important to not dilute your signals specifically in rodent, because then you may actually not see real functional connectivities between compartments.
Yeah, I mean, it's one, I think this is really because of the advances in a lot of instrumentation, but then two, you're mixing these dreads. So I mean, this is an example of a bunch of different fields coming together.
Oh yeah.
And really allowing us to produce a new cutting edge, right?
Oh, so absolutely, that's exactly it. And that, I think the beauty is that this is where the direction of neuroscience is going. And so we are, like I said before, this whole notion of going back to basic neuroscience, it's happening right now. And it's really cool because it basically is starting, with a tabula rasa, just a blank slate. We're just saying, okay, let's just re-envision the brain, and then let's try to figure out what's going on. And, some people are asking it in these type of ways, that they're just like, well, let's forget about those rigid lines from before that were established by like these particular, let's just start new. Let's look at it in a way that kind of makes maybe more technical sense. I
love this animation, by the way.
those moving images right there are just the type of images we can get with a scanner like ours, which is it's a 9.4 Tesla over at UNC. We call it the CAMRI, the Center of Animal MRI.
Okay, no, that's really confusing because we're talking about Teslas and Camrys and Civics. No, I'm kidding. So the Tesla, just for clarification, there are not 9 Teslas in the basement of Marsico at UNC. It's a magnetic measurement of magnetic field, right?
Yeah, exactly.
So just for clarification, in case
just a clarification.
Yeah, there's not just a parking garage down there. So yeah, I mean, let's talk about what actually happens when you activate the locus coeruleus. So you're activating this locus coeruleus. We know it's be important for noradrenaline, norepinephrine release. Doing that with dreads, and then you compare that in an animal with the dread, and then an animal without, right?
Yeah, I think this is one of the coolest things. I remember I was talking to a person that just does, I think, like what was it that they study? I think like culture studies or something like that. And one of the coolest things, they look at a figure like this and they're like, oh, this is one animal. Like, no, we could literally group our animals, like because the brain, we could actually we warp the brains and put them into the same space, and then we could actually compare that way. And so when we look at this, these are actually comparisons of a huge, like a study population, and the differences between a study population.
So this
expressed
is average
as one brain. So it's like one average. Really cool. Yeah. And so one of the things that we see here is, like I told you before, norepinephrine is a very potent vasoconstrictor. And that's exactly one of the things we saw with just cerebral blood volume. So we see that in many systems, it just completely constricts the perfusion in those areas. So that's why this is blue. So we
less
see
blood flow, right?
Negative. What?
So it's less blood flow, right?
Less blood flow. Yeah, less blood flow. So we're basically all the vessels with tone are just, kind of having smaller luminal diameters. And one area that's really interesting, if you look, the areas that are actually in white that haven't changed are some of the sensory areas. So these are, where you have, the motor sensories and somatosensory and stuff like that. So those aren't really changing, which kind of gives you an idea of maybe we're modulating a system. So this is, when I told you before, this thing bathes the entire brain, but that doesn't necessarily mean that everything responds in the same way, which is very evident here. The other slide right there is PET FDG. So this is our glucose uptake. So as I told you before, traditionally the default mode network is really studied in these two ways, using just a form of imaging metric from fMRI, and then the other one is using glucose metabolism.
Mhm.
And one of the things we see is when we activate those neurons, so the bottom right shows you that it's all blue. It's more blue in particular. So we actually have less glucose activity. And so this is really interesting, because one of the things that we know is that norepinephrine could actually alter how we utilize glucose for short periods of time. So it could actually act directly on astrocytes. And instead of utilizing glucose, they actually will go and utilize their glycogen reserves. So astrocytes, if for those listeners that don't really know much about this, are like this mediating cell, a glial cell, which is actually responsible for most of the uptake of glucose from our blood vessels. And then it converts that glucose to be able to send it to neurons to use as their energy source. And so they're kind of this intermediate cell that is kind of the feeding tube for our neurons.
It's one of the many things astrocytes do. They are just an incredible cell type. Like, definitely understudied, underrated. I love them. So that's my little
they're really, really cool. But this, in the context of like metabolism, it's really important. But so they're also one of the few cells in the brain that actually have metabolic reserves, and they do it in glycogen, which is just basically glucose and like tons of, a big molecule, and then you just cut it open and pull the glucose off and just send it that way.
What's tiny cool little about that, sugar cubes.
Yeah, exactly. But what's cool about that is that type of metabolism is much faster. You could go much, much quicker. And so part of the reason we suspect it uses is that mechanism is in case you need those like immediate responses. And let's say you have like a cut or something, and like you're not getting that much blood flow going up to your brain. It's like it's still a way for you to engage really quickly, blood flow or not. And so this is a cool mechanism which we're seeing right here in action with glucose. And so if you go to the next slide, that in a way kind of indicated to us that all these systems are actually suppressed. And so typically, if you were to look at it in the historic way, we would just say, well, because the fMRI and the glucose have gone down, that must mean that our neuroactivation has gone down, that it's actually a inhibitory activation of an inhibitory signal. But that's actually really not the case here. And so one of the ways we could actually study that are these methods. So one of them is called REHO. Another one is the amplitude of low frequency fluctuations, and the other one is through functional connectivity. So one way that you look at it, so REHO is just giving you your maps the regional synchrony of spontaneous neuronal activity. ALFF really looks at only the low frequency oscillation signals, which is associated also with spontaneous neuronal activity. And then functional connectivity, basically you could take 2 points in an fMRI map, and you're just basically looking at the trends of their activity. And so those that have extremely correlated activity are typically connected, right? And so this is kind of, and the thing is, neurons can change how connected they are and how disconnected they are. And so this is a cool metric because you can actually see if something gets more functionally connected or are kind of like clustering together and firing together and like communicating together or less. So these metrics are other ways that you can actually look at data. And if you go to the next slide, what's cool is here is the view under, it's kind of like pulling up the veil from what we've originally done in the past. Here we see that in the REHO, we actually have an activation. So it's the reverse of what we're seeing regarding the signal. And so by looking at a different metric that's actually more associated with the neuronal activity, we're starting to say, hey, up in the frontal area of the default mode, in these kind of frontal areas like the anterior cingulate, we have this high activation and high synchrony of cells firing. In ALFF, the same thing. We're seeing these different areas of the default mode actually have activity. changes that are different than when they don't have norepinephrine release. And then in the functional connectivity, the third one, they're the same. We start looking, when we just look within modules, we see that in the frontal, there's a higher within module connectivity. And then between two of the areas, between the frontal and the association, we're also seeing higher connectivity. And so this kind of sends up kind of these red flags for how we study the neuromodulatory signals, because we really need to focus in on the actual neuronal component of this. Sometimes if the module itself, the neuromodulator itself, can actually change things like glucose or the cerebral blood volume or bold signals in general, just based off of perfusion. So this is cool because it kind of unmasks the signal that would have kind of historically maybe been overlooked if you don't go a second layer into analysis.
So like I was talking about, this intersection of many technologies, not only is it the DREADS in terms of biotechnology, also the imaging in terms of engineering. Third component here is computing, right? So it allows you to very rapidly in these large data sets kind of, and also I should mention probably an unbiased manner, to look at these three separate models and say, okay, we do see changes in blood flow to the brain, which again could be a proxy for activity, but you don't stop there. You go another layer using these computing techniques to say, okay, yes, there's a wholesale change in cerebral blood volume or blood flow in some of these areas, but then there's also these specific smaller pockets that are changing in not necessarily the same rate, but they're changing at the same time in the same direction. Would that be a fair thing to say?
Sure. Yeah, we could say that. Could see that. Yeah. No,
and so
it's really cool because it puts a big cautionary tale in a way of, like, in the past when we used to report data, we'd just be like, oh, fMRI change, and we'd just show you that top corner. And so if we were to have said that, we would have completely missed the point of this whole paper. And so what's cool is we were fortunate enough to get a network scientist who's Li Ming, who's on this paper. He's one of the co-authors with me. And so he really helped us really understand network neuroscience in a much clearer way. But as you said, that component of the computational side, and especially now with machine learning, is just blowing up in this particular field. And all those things are extremely novel, and they're really helping us parcelate things that, in the past we would have never caught.
Allowing you to see the trees through the forest.
Exactly.
Essentially.
Yeah, absolutely.
And all the nuances.
Absolutely.
So this is this is really big. I think, like, where do you see this kind of this information guiding pharmacological development, guiding drug development, or systems, even, kind of in terms of a systems perspective, where do you see us thinking about the locus coeruleus in the future?
Yeah, so now from this, we actually know, hey, we know that it's actually modulating neural activity in these systems, and specifically in the default mode network, which we kind of suspected. In humans, we can give drugs, pharmacological drugs, and it kind of, everything kind of hinted that that's what's happening. But this actually really, because we're using a model that's very selective, it kind of gives us more of a causal association here, which typically you can't really do in human. And so this at least lets us know where norepinephrine is really, what areas neuronally it's really modulating. But what's cool is the stuff in, I think, the next slide. So this is us verifying this slide. So this slide, what's cool is you could do what's called dynamic causal modeling. And so this basically says, let's say you have 3 findings. In this scenario, we kind of did. So we have over on all the way to the right, we knew that within the functional connectivity within the frontal module got enhanced. The one in the top is between the frontal and the association. And then we ran a different type of study that really let us understand kind of the causal nature of if you look at associations between two particular things, is it better, stronger, or weaker? And what we found out was that we had this kind of causally reduced control from retrosplenial hippocampus to association model. So think about it as like releasing the gas pedal, basically. So it's a reverse type of thing. So we actually lose control there. And so with that, we were able to build this type of model in which we could say, okay, if we think of the retrosplenial hippocampal module as kind of the model input, and then we think about the frontal DMN module all the way to the yellow on the side there as kind of what we're changing. So here we're changing like an association with cognition and the type of outputs. Then if we follow this order, can we actually prove that one is in charge of the other? And that's exactly what this does. This model with these graph theory actually let you do that. I think if you hit twice or three times, you'll uncover some more text here. Yeah. So basically what this really tells us is that if you shut off the information flow from one of these modules to the other, so this is what norepinephrine does in this particular order. So it really starts in that retrosplenial hippocampal area. So it's kind of more like a top-down type of adjustment. And it uses this mediator, which is the association cortex, to signal that frontal area that we saw those changes in the ALFF and REHO. And so this kind of really gets you closer to what you should be targeting, which is that connection of norepinephrine towards that retrosplenial hippocampal area. Because if you hit there, you can adjust all the downstream parts. And so what we can then start viewing is if we look at things like chemogenetics in the future, is we've maybe find the cellular population that norepinephrine's really acting on to do this, and then target those cells. And that could help us really with this particular distinction between the two.
So in any of the diseases that you listed earlier,
exactly,
like even Alzheimer's disease, this has implications where people are losing focus. So,
well, not only that, the reason they're losing it is in Alzheimer's and Parkinson's disease, they actually degenerate. The locus coeruleus cells are degenerating. It's actually the largest group of cells that disappear in people with Alzheimer's. So it's like, we always think about like the hippocampus and Alzheimer's and the substantia nigra and Parkinson's disease, but actually the largest system is the locus coeruleus of loss, of degeneration, is actually the locus coeruleus.
And in so the timing, it's one of the first. Yeah.
Oh yeah. And part of the reason you want to study those particular regions is because those are all those other side effects that you get are like the lack of attention, GI issues, like all these particular things are associated with that particular component. So if you can help alter some of that, you could probably help relieve a lot of symptoms and stuff in people. So
yeah, I love it. I mean, this is really foundational work. You're kind of not necessarily doing a clean slate, but you're giving a fresh perspective on network analysis. And I think that's just so important, as well as like the implications of the locus coeruleus with attention. I hope a lot more comes out of this as well. And I'm sure it will.
Yeah. Thank you so much.
Yeah. So Esteban, I got to ask, last time we talked was in person on UNC Chapel Hill's campus.
Sure.
Obviously you've done great work in the She Lab. And if you want to shout out others, please feel free to do so. But
the UNC Lab. So if you want to go visit them, they're at camry.org. They are a fantastic group. They're also a core service facility for not only UNC, but all types of experiments throughout the US. So if you want something done, anatomical imaging, DTI, functional imaging, this group is fantastic. It's one of the premier groups in the world for rodent imaging. So definitely a shout out to them for sure.
So what have you been up to since? I know graduation can be an adventure, but sounds like you're in a new world now.
Oh, absolutely. So right now I took a job with Charles River Laboratories. And so many of you who are scientists may know us from just breeding animals in general. So we you know, I think it's like for every 5 animals, 3 of them are produced by Charles River in research in particular. So it's like that's where a lot of people know us from. But what we have is we have a branch that also does all the preclinical kind of screening. And so I'm here in Michigan right now at our Matawan facility, which is one of the largest facilities that does toxicology and pharmacology screening of all drugs prior to them going into clinical trials. So this is the preclinical phase of drug screening. And I am a study director with the molecular imaging group. So we are primarily taking these drugs that are coming through the pipeline. Some of them, right now the field is so new in a lot of ways. So traditionally it would have been just all these small molecules hitting receptors, the lock and key idea. Now it's just all types of things. It could be RNA, like the COVID vaccines and stuff like that. It could be oligos. It could be large molecules, like antibodies,
other proteins,
all that stuff. So it's really cool. Or implants. It could be all types of stuff. And so we're basically whatever we could put a radioisotope on, we could start studying kind of the pharmacokinetics and the biodistribution and the clearance using PET imaging or SPECT imaging, depends on what radioisotope we do. And so, I have a fantastic team out here, and we're doing that type of work right now, which is really cool because it's in the pipeline of, drug development. And so it's stuff that the end goal is for humans. It's really, we're trying to, relieve a lot of hardship and stuff like that for people with chronic disease or acute diseases, which is fantastic to be in that type of work right now.
Yeah, definitely. I think like we had kind of talked about throughout the episode is, there's been so many fundamental advances in basic science, and like neuroscience reinvented itself. And that is huge thanks to academia on that.
Absolutely.
Like, obviously academia was the driving force, but now there's this massive toolbox available for industrial partners. And as much as pharma companies get a bad rap, like they're still there for the purpose of solving human diseases, human ailments, and improving quality of life, right? So to be able to see like these technologies applied so vigorously, I mean, that's the huge draw to industry jobs to me, right?
Oh, absolutely. And I think the other bit is the component of machine learning. And so typically in the past, like I told you, a lot of it was small molecule stuff. And so there was only so much you could do, because it was also, you need to have intellectual property rights. And so you can't have something that already has been developed because you don't own the rights to it. And so, they kind of got to a point that they kind of used all they could. They found all the molecules they possibly could. But the reality is machine learning has changed that as well. All of a sudden, there's a whole bunch of new small molecules that we're seeing that it's just, machine learning has really changed how those models actually spew out new molecules as well, which is really cool. Yeah, that are more selective. That's the other bit. It's like we're now learning like depression. Like there's some papers you go out and you're seeing like, oh, they identified this other cell group that have these orphan receptors that don't express anywhere else. That's like the holy grail, right?
Yeah, for targeted therapeutics. Exactly.
Okay, we can just target this one thing. Yeah, of course. That's like, it's really interesting to see what's down the pipeline right now for drug development. And it's a fun time to really be in this field.
Yeah, I mean,
I guess
you don't have to.
We can transition to you a little bit right now and what you've been up to, Connor.
Yeah, so I mean, I would definitely do for an update episode just for the listeners who have been following. But for my own part, I had kind of a similar experience. I saw in academia like this massive focus on tool development, and it was, I mean, it's really incredible what has happened in the last 10 years alone on that front. So I think industry is now ready using either these huge screening approaches or like these machine learning approaches that you talked about using computing power, and then also these big data sets where we can get, thousands, hundreds of thousands of patients and their information to really track like what's going on throughout everything. And so, yeah, I'm 3 months into postdoc here in California at Alcahest, and essentially Alcahest is focused on like the proteins that are typically found in blood, and how those change throughout aging, right? So we know that from some previous experience, again, shout out to academia at Stanford, is that a lot of proteins are changing just in the blood alone. So this is, we're taking a step back from the brain and saying, all right, what's happening across the body throughout aging? Some proteins go up, some proteins go down, and that's an oversimplification, of course.
Sure,
But,
of course.
we want to know like what are the difference makers between the blood of like a young mouse versus an old mouse, or hopefully extrapolate that to humans. And there should be a lot of things in there, like potentially some of the factors that you're testing at Charles River.
Sure.
A lot of those are proteins, and we know that proteins can make valuable therapeutics, right? So
Absolutely.
basically part of my job is getting the toolkit ready to house a lot of these experiments. And so that's really exciting, and being able to see what are the steps that, like, how many safeguards there are just to make sure that we're doing the right thing even before it gets close to humans at all. I mean, that's the kind of the joy in it, just to make sure that everything is ship shape and we're asking the right questions. But then also being so close to the clinic compared to how I was as a grad student, I think is really rewarding, because I think a lot of grad students join because they want to make a difference. And they absolutely are. But for me, it's very gratifying, I guess I would say, to be a little bit one step closer.
Closer. Yeah, I think that's an important thing that you need to ask yourself as a researcher is how close do you want to get to the outcome? Because there's some model, some labs that just only work more in, kind of the basic side and basic science side. And so you may not necessarily ever get to the disease,
right?
Or if you may be working in the disease model and you've never seen a human patient. I've once worked actually in a Parkinson's disease laboratory, and I asked my colleagues how many of them have seen Parkinson's disease patient, and few of them had. And so having that type of understanding of yourself and like how close you want to get to it, I think it could be more rewarding for you to be able to see kind of the end goal, like the insight, if you are more oriented towards like the towards the drug development and therapeutic targeting aspect versus maybe the basic science.
For sure. Yeah. And it's all about finding the balance. Like maybe I go back to basic research at some point or something like that. And I really like the questions I'm asking now, and I'm gonna for sure be able to hopefully talk about them more in the future as I develop. Yeah. Cool. Esteban, this has been great. Thank you so much. I do want to like just ask a little bit about you. So it's Saturday. Like, what are you up to in your free time when you have it?
Well, so I'm also kind of new out here. So I'm in Kalamazoo, Michigan. And so I'm more into like mountain biking and hiking and,
Nice.
playing board games, stuff like that. So that's primarily my free time, what it looks like. But yeah, as you can imagine, it's also long days as well in these type of laboratories, especially more in director position. It's you have to make the final call on certain things. If an animal's not doing well or something like that, you could get that 1 a.m. call or whatever, so you may have to come in. So but yeah, but now it's there's snow on the ground. I don't know what I'm gonna do yet. I think I'm gonna be doing some skiing or something like that. Who knows?
Definitely.
We'll see what happens. It's been a while since I've been in a more four season style climate.
That's right. Yeah, I tell people we typically just get like 2 seasons.
Yeah,
In
that's where
Chapel
I'm
Hill,
at
yeah.
in North Carolina. Yeah, now even more so for you now,
Oh, I got 1. Yeah, that's it.
Yeah, one beautiful season.
Yeah, it's pretty hard to complain about, I gotta say.
Yeah, I know.
Nice,
Exactly.
man. Well, I mean, if there's anything else you want to mention, I think now's definitely the
No,
time.
I think go out. The papers in bioarchives. You could go find it on Twitter if you want to from
I'll
the
link
Camry
it.
site. Yeah,
Yeah, I'll
from
link
Camry
it for sure.
site.
And then if it
Um,
gets updated in the near future, I mean, not
so yeah,
regardless, I'll have
yeah.
it
And
updated.
we
But
are making changes. So in bioarchives, anytime you change it, or as it gets published,
mhm.
then we'll make those changes as well. So cool. Yeah, that's just about it. And yeah,
Awesome,
shout
man.
out also to Manasmita. She was also one of the co-authors as well. So we had 3 co-authors here. And the She Lab in general, they were really a fantastic place to really learn this particular field. And when I got there, it was just in its infancy. The
Mhm.
reality is like we were really globally starting to do mouse work. And so we've really pushed the technology in so many ways in this particular paper. And so it's really cool to have been able to have those opportunities. And UNC, as we
Mhm.
both know, really offered opportunities that you sometimes can't really find in an academic setting. And everyone's just so excited to be involved in that.
Yep. Shout out to UNC for sure. Yeah,
For sure. Cool. Okay, well, thank you so much. I
of
really
course. Yeah.
appreciated this
Mm.
interview. This is great.
Very glad to have you on, and I'm sure we'll be in touch in the future.
For
Thanks
sure.
all for listening, and take care. Have a great holiday season.